you very much for this introduction, and thanks to all the organizers for inviting me here to this panel. I'm Robert Fischer. I'm coming from Omikos Therapeutics, a company, small biotech company based in Berlin. And I'm happy to give you today a little bit short, quick overview on our first data that we received from our 2A trial in mitochondrial disease, which of course would not have been possible to achieve without a lot of support from many scientists here which are in the room, Thomas Klopstock and his team, but also many of the Italian investigators. So introducing our lead molecule, OMT28. So OMT28 is a small molecule acting as a biased modulator of this sphingosine 1-phosphate receptor 1, which has also been linked to regulation of mitochondrial function. It biases this receptor to signal via GI to increase activity of major deacetylases, so sirtuin-1 and sirtuin-3. And as a consequence, we see increased activity, in particular under stressed conditions of complex 1, but also other mitochondrial OXPHOS complexes, leading to improved ATP production, reduced ROS production under situations of metabolic stress. And we also saw upregulation of NAD+ levels and GSH levels in preclinical assays. And we also observed in preclinical assays that via PGC1 alpha upregulation, we saw increased mitobiogenesis in some of the cellular assays. Overall, of course, we hope that this helps to improve helps to reduce progression of the disease in our patients, improve functional exercise capacity, and reduce fatigue, and overall improve the quality of life of our patients. So what we did with major input from the investigators, a 2A trial, which was an open-label study, started with a 3-month non-treatment period, and then followed by a 6-month treatment period. We had rather broad inclusion criteria, so including patients with mitochondrial tRNA point mutations, but also mitochondrial DNA deletions. We were broad with regard to the clinical phenotype, so we said they need to have either skeletal myopathy or cardiomyopathy, and they all needed to have an increased GDF-15 level above 1,200. Endpoint-wise, of course, we looked majorly for safety and tolerability, but also for major biomarkers and all the functional endpoints that you know all very well. With regard to the population, I think there are no major surprises. So we got an equal distribution of males and females. Of course, most of our patients had the M3243A2G mutation, but we also had patients with mitochondrial DNA deletions and also 2 patients with MERS syndrome. Otherwise, I think no big surprises. Maybe we had a bit more of the cardiomyopathy, so cardiac manifestation in our population based on the inclusion criteria. Safety and tolerability, we can make it short, that was absolutely confirmed, so no issues here, also no special adverse events of special interest. Cardiac safety was confirmed, so I think very nice results here on the safety side. Of course, we looked on many biomarkers, and I can say what was most interesting for us were changes that we observed in NAD+, NADH levels, but also GSH, GSSG levels. You see here on the left-hand side increases of NAD+, NADH ratios in the entire patient population. However, when we did a split between patients that clinically responded versus patients that clinically did not respond, then we saw that this increase in this ratio was mainly driven by the responders. And the responders here were defined by a combined improvement in 12-minute walk test and 5-time sit-to-stand test, or they needed to improve in the patient global impression of change score. The same was true for GSH, GSSG levels. You see this nice increase here in the entire patient population. However, let's say this increase is in principle majorly seen in patients with clinical benefit, pointing overall to a reduced oxidative and reductive stress in these patients. Of course, we looked also on motor function tests. You see this increase here in the entire over the entire study period. Again, this was significant in our responders, and we also saw this improvement of the 5-time sit-to-stand in the responders. When we looked for correlations between, for instance, GSH and GSSD ratio, we saw in the responders a very nice and significant correlation, which makes us confident that what we see here is not a by chance finding, but really a kind of mechanistic link between these, between the motor function and the biomarker. And by the way, this analysis of these biomarkers was done in collaboration with NUTMATE in Finland. So what we want to do is now we are still about to complete our full data analysis. We want to conclude this now in this next weeks to come. We also implement AI-based advanced biostatistical methods for a more in-depth analysis of the data set. So that's also some kind of new experience for us. We plan then for a confirmatory phase 2b/3 style study. And of course, also here we will need input from all the investigators and scientists on what is the best design and what are the best and most optimal endpoints to select for this next very important study. And we just heard it, so of course we will seek advice from the regulatory authorities. So we plan to have meetings with the FDA and the EMA in this year. So overall, thank you very much for having me here, and happy to discuss all this.